right, welcome everyone to our last chapter that we'll be covering in this course. So we're going to start our reproductive system bit of the unit today. Now, before we get going, I do want to just start with a disclaimer. I know a lot of you are going to go out into various health professions, and we're in the end of April here. We're keeping our study of the reproductive system to the basics, the typical, the average that you'll see out in the world. But when you meet actual patients, there are a lot of other things that might be going on in their lives. So my goal for all of you is to kind of keep all of this in context. So when we use the word male and female in these slides, we're talking about typical biological sex, but there's a bunch of other things that affect people's experience of the world. And what I can also say to you is that there's a lot of cool, weird stuff that can affect people's biology as well. This is a topic I care a little bit extra about because I have a friend who discovered when she was 16 that she didn't have a uterus and had no idea. So I just want you to know that there are a lot of people in the world whose bodies aren't quite the standard that we're about to talk about, standard male or female. About 1 in 100 people have something different going on. About 0.06% of people aren't even XX or XY. Some people have cells that could have different genetics in each cell because of these concepts of mosaicism and chimerism, which you might find out about in a genetics class. We will not be covering that here. Okay. And about 1 in 1,500 people are born with genitalia. Atypical enough, you have to call in a specialist. About 1 or 2 people in 1,000 has to have surgery to quote unquote normalize genital appearance. 1 in 6,000 people is born with an absent vagina, and some people are born with ovotestes, so gonads that are simultaneously an ovary and a testis. I'm not going to be testing you on any of that, but I do want you to know that that's out there, that exists, and there's more to dive into if you end up going down this road in your future careers. Okay, so that said, we're going to do our basic overview of reproductive physiology. Right. So we're going to be thinking about what are gametes and what's their role in sexual reproduction. We're going to be thinking about the role genes play. How do genes get sorted, packaged in the formation of gametes? We're going to be thinking about components of the reproductive system and what happens after fertilization through the course of someone's lifespan. So we're going to start out with the role of our sex chromosomes. And to do that, we need to think about what they are. Okay. So when we think about our reproductive system in total, we're thinking about all our organs that are involved in the actual physical process of mating, the process of gametogenesis, which is where you make gametes, so sperm and egg cells basically, and other functions that are involved in producing offspring. So the key to this is going to be our gonads, right? Assuming everything's working normally, males are going to have testes, females ovaries, and these are produced from the same type of tissue as we look through fetal development, as we will in a second. Okay. So the major function of either of those is going to be to produce the gametes, either the sperm or the egg cells, as well as to secrete sex hormones. So we'll be thinking about our androgens like testosterone in particular, and also estrogen and progesterone. We'll discuss some accessory reproductive organs as we move forward, but we're first going to be thinking about really the sex differentiation part and the sex hormone part. Okay. So our testes are going to produce androgens like testosterone. Ovaries typically produce estrogens and progesterone. What's kind of cool, kind of interesting is that if you're out here sitting in the audience or me, right? Most people have both, not both testes and ovaries, but you should have both androgens and some estrogen and progesterone in your body. This is about proportion when we look at males and females. Males tend to have more androgens because they have testes. Females tend to have more estrogens and progesterone because they have ovaries. But your adrenal glands, everybody's adrenal glands also produce testosterone, and testosterone can be converted in different tissue types, like in fat, into estrogen. So the picture isn't as black and white as it might at first appear. These are going to also affect our accessory reproductive organs as we walk through them. When we talk about organs of the reproductive tract, we'll be talking about things that get transported by, or things that transport our gametes. And we'll be thinking about glands that are going to be secreting fluid into the reproductive tract. So we're going to start shortly after fertilization. So it's worth taking a peek right now at how we get there, and we'll circle back to it at the end. But when we talk about the act of mating, you might also use the word copulation. What we're focusing on here is sperm being deposited into a female and an ovum that might be fertilized. Okay. So after fertilization, which we typically refer to as pregnancy, but a more formal word we might use is gestation. We're going to use two separate terms, but flowing into each other. So if you've used the words embryo or fetus before, you should know that from a physiological perspective, those have specific meanings. So for the first 2 months, we're referring to that as an embryo. And then after 2 months, we're at this kind of fetus level. So after about 9 months, we experience parturition, which is just another fancy word for birth. Okay, so what's happening in the in-between here? And one of the things that we're going to be tracking to understand the reproductive system is how do you end up with an adult male or female reproductive system? So this process is going to start at about 2 months into a pregnancy, a little bit before. We're going to start to determine whether we're going to develop ovaries and go down this female route, or we're going to develop testes and go down this male route. So we refer to this process as sex determination. Okay. Now, the key to sex determination is the Y chromosome. This is kind of the only thing that matters here, because we're going to see that any embryo that doesn't get this signal develops into a female fetus, right, for any reason. All right, so we're looking for a Y chromosome at this point of sex determination. And the reason we're looking for a Y chromosome is it has this specific gene on it. Okay, so this SRY gene, or sex determining region of the Y chromosome, is the specific thing that tells your body to make testes. So if we have an SRY gene, we're going to produce testes. If your SRY gene is absent, doesn't work, if you just don't have one, you're going to produce ovaries. Okay. So this SRY gene is able to do this because it codes for another molecule called testis determining factor. So we're thinking flowchart wise, we got a Y chromosome. On the Y chromosome, we got an SRY gene. The SRY gene codes for this protein. TDF is what we'll often see this referred to as, testis determining factor. Okay. So if you have testis determining factor, you're going to develop testes. And if you don't, you're going to develop ovaries. Okay. So it's these hormones that are controlling sex differentiation. First, this TDF, and then the hormones produced by functional testes. Okay. So for our first 2 weeks, for sure, our embryo is sexually indifferent, and it's going to have the precursors to both of our tracts. So we're going to have Wolffian ducts that are a precursor to the male reproductive system, the male reproductive tract. And side by side, we're going to have the Mullerian ducts, which are the precursors to the female tracts. And then slowly, we're going to start to produce these changes if we have a Y chromosome present. With this SRY gene starting to get red, we're going to start to produce testes. Okay. So when we get these testes online, right, once we actually form a testis, it's going to start to secrete 2 hormones actually that we're interested in at this point in time, right? Usually we just think about testosterone in adult life as our androgen, and testosterone is important here. So as soon as we have testes, we're going to produce testosterone, and this is going to stimulate these Wolffian ducts to mature and to continue to grow. But if we only had testosterone, we wouldn't be done because those Müllerian ducts are still there, right? These reproductive tracts, these potential reproductive tracts are developing in parallel. So that's why this other hormone is important too. So the testes are also making this hormone called MIH, or Mullerian inhibiting hormone, okay? And what that's doing is it's telling the Müllerian ducts, so the ducts that would usually develop into the female reproductive tract, to degenerate, right? So we both have to develop the Wolffian ducts on purpose with the testosterone and kind of kill off the Müllerian duct that was there just kind of waiting to see whether we'd get a signal from testosterone or not. So if no testis forms for whatever reason, then we're going to see lack of these hormones, which means the Wolffian ducts will degenerate instead, and the Müllerian ducts will be the ones that keep growing. Okay, so we're going to take a look at this picture. Okay, so what we see here in yellow, this is the primitive gonad. All right, so early on in embryonic life, in this sexually indifferent stage, you've got just a gonad that's going to eventually become a testis or an ovary. Next to it here, we can see that Wolffian duct. All right, so here's a Wolffian duct. All right, so if the Wolffian duct ends up developing, all right, that's going to take us down the male route. But at the same time, we're also ready to build a female body. So right next to that Wolffian duct, early on, we have this Müllerian duct. Okay. So this is our setup, ready to build kind of any reproductive tract we want early on. So what we've just spoken about is on this, for me, left-hand side. Yeah, left-hand side. All right, tracing that male development. So you can see how this yellow tissue, our primitive gonad, turned into a testis. All right, so we can see that right here. Okay. We can see how our Wolffian duct here, all right, is now surrounding that testis, basically. All right, and we've produced from the rest of that Wolffian duct our vas deferens. And we can see a seminal vesicle forming here to the side. All right, so that's happening around 8-ish weeks for this embryo, and then speed running through. Eventually we end up with a testis that looks like this, a longer curling vas deferens, coming in to where we have our seminal vesicles, dumping in at the same time as we pass through the prostate, and ultimately out the urethra and the penis. Okay. So that's what happens on the male side. So we had a Y chromosome, we had an SRY gene. SRY gene made TDF, testis determining factor, which is why we made this testis. And then as the testis produced testosterone, we beef up this Wolffian duct. And because we also have MIH, Müllerian inhibiting hormone, we killed off this Müllerian duct. All right. So if we don't get any of those signals, we go down the other side. All right, so instead of forming a testis, this gonad still develops, but now it develops into an ovary. We don't have any testosterone coming from an ovary, so the Wolffian duct degenerates, right? So that's why this gets all pale and dotted in the image. And instead, the Müllerian ducts continue to grow. So we can see that Müllerian duct turning in to a uterine tube, heading towards our primitive uterus here, and eventually we get our full-fledged kind of adult form where you've probably learned all the parts in anatomy lab, but we'll go over some of them again as we get to the reproductive anatomy in this portion of the course. Okay. So that's really the first piece that we need to think of when we think of how do we get to normal adult function of the reproductive system, right? So first we need sex differentiation to happen. We need to produce a male reproductive tract or a female reproductive tract. Then we need puberty to happen. Usually this starts around 10 to 14 years, variable in sexes, variable in people. But what puberty is sexual maturation. So when we think about this from a reproductive organ standpoint, what we mean is that we can now produce functional gametes, right? So you produce sperm and ova that can actually get fertilized or fertilize each other, right? We are also going to see secondary sex characteristics developing around this time as well, right? So as we see this kind of general hormonal shifts start to happen in puberty, we'll start to see changes to voices, changes to, I don't know, breast tissue, these kinds of things happening along with this maturation of the reproductive organs themselves. So during the sexual maturation process, in particular, it's going to be noticeable that the female reproductive system starts to cycle. So women start getting their periods, right, during puberty. So we're going to be tracing, when we get to that side of the equation, all the different hormones that you'd expect to see across the course of a month if you have a female reproductive system. Okay? So that's going to start during the sexual maturation process. It does end eventually. So during menopause, we will see the loss of female reproductive capacity. On the male side, we don't have the same hormonal cycling that we see in the female side. And we don't really see an exact analog to menopause either. Males are going to retain the ability to produce sperm throughout their lifespan. Okay. So we are going to stay on the male side first. We're going to be thinking about how we produce the male gametes, the sperm, how they mature, and how they're delivered. Okay. So running through some of the anatomy that I suspect you've seen, but let's just make sure. All right, so we've been talking about the testes. All right, so we have the testis down here in the scrotum. All right. We have our epididymis sitting on top and looping around. All right. We have our vas deferens. It's ultimately going to carry sperm produced in the testes into the body. All right, so looping around, looping around, looping around. Behind the bladder, we're going to see the vas deferens meeting up with the seminal vesicle, which is going to add some fluids. Okay. We then see that we've got this ejaculatory duct going through the tissue of the prostate, meets up with your urethra coming from the bladder. Right. So now we have the urethra continuing out through the erectile tissues of the penis and ultimately out of the body. Okay. So we'll be tracing through what happens as we go through these different areas and as we pass each of these glands. But first, we're going to be focusing on what's happening in the testes as we're producing sperm. Okay. So the testes are the male gonads. If you were to slice the testis open and zoom in real far, you would see that a testis includes tons and tons of little compartments, right? So what we're looking at here are the 250 to 300 compartments containing seminiferous tubules, right? So these curly ramen-looking things on this image, those are seminiferous tubules. And then if we take a cross-section through an individual one, we're going to see this region where we have sperm production happening. Okay. So this is cut through, essentially, one of those seminiferous tubules. All right, so we're going to pay attention to a couple different cell types here. So in purple, kind of around the edge, here we see our Leydig cells. Right? So these Leydig cells are a type of interstitial cell, so like between tissue. And these are the cells that are actually making the testosterone. So these Leydig cells kind of out and around those seminiferous tubules. Actually forming the walls of the tubule, right, we have these Sertoli cells. So these Sertoli cells are a specialized type of epithelial tissue. And these tissues are going to support sperm development. All right, so if we zoom in even farther here, we can see our Sertoli cells here. All right, we can see that we're going to have a tight junction. We'll talk about that in a second. That's going to separate out what's happening kind of towards the outer edges, an area we call this basal compartment, versus what happens in our luminal compartment, which is the region that's in towards the middle, where we have eventually our sperm collecting. Okay. We might also notice that out around the outside, we also have some smooth muscle, right? So that's not one of the types of cells that we're tracking, but we are going to be thinking ultimately about how do we get sperm out of the testes. So it's helpful to realize that there is a little bit of muscle here. Okay. All right. So eventually, that smooth muscle is going to propel the sperm through the process of peristalsis, which we actually spoke about last time for the GI tract, right? So same kind of process here. We're going to have essentially squeezing little bit by bit of the tube in order to move sperm forward. Yeah. So it's also important to realize that we are protecting the sperm as they develop within the testes. So like we had a blood-brain barrier up in the brain, we actually have what we refer to as a blood-testis barrier down here in the testes as well. Right? So that's going to be formed because we have these really tight junctions separating out the inside of the tube in this luminal compartment, right, from the contents of the basal compartment kind of out towards the edges. We can also see what these look like in our real histological slide, right? So you can see a Leydig cell producing testosterone out here, and then we can see these cross sections through our seminiferous tubules roughly here. Okay. So when we're tracking through what's happening here, right, our sperm are going to be developing and kind of like descending. I would think of it a little bit between these Sertoli cells. And the Sertoli cells are going to be supporting their development. Okay. So this means they're going to be secreting some fluid to put into the lumen, so luminal fluid, right? Because we don't just have a bunch of dry sperm with nothing else, just clustering in the center of the tube. We've got a little fluid. They're also going to be secreting something called androgen binding protein. So this is going to kind of buffer things out a little bit. So it's going to be helping to maintain a steady level of androgens in the lumen. So here in where the sperm are ultimately collecting by binding some of this stuff to this protein. The Sertoli cells are also our target cells for testosterone and something called FSH, or follicle stimulating hormone, which we'll talk briefly about here, but loop back and talk about it significantly more when we talk about the female reproductive system. And kind of at the end, we can draw some parallels between how these hormones function in each. But we're thinking about these cells as secreting some chemicals, essentially, that are going to be stimulating spermatogenesis, so stimulating the process of actually making sperm. Are also going to be secreting another hormone called inhibin. So when we look through our flow charts of how all the hormones are affecting each other, we're going to see that inhibin is going to be basically a negative feedback hormone acting on FSH. It'll make more sense when we ultimately like draw out these pathways together. Okay. But as we're running through the anatomy, we saw them these pieces on the slide, the cross-section, right? But our external genitalia here in the male reproductive system is going to be the penis. We might also refer to it as the male copulatory organ. For being formal, we're referring to its purpose as sperm deposition, okay? So at rest, our penis is flaccid, so floppy. During sexual arousal, undergoes erection. So what's happening here is we're seeing a lot of blood flow to the penis increase. The blood gets stuck in the erectile tissues of the penis, which causes it to harden and become straight. And this happens through to ejaculation. When sperm get ejected through the urethra, we trace their path on our anatomy slide. Other pieces of the external genitalia that we might notice, we got a scrotum roughly suspended beneath the penis where we house the testes. Part of the reason you need a scrotum in the male reproductive system is the temperature requirements for sperm development. Sperm actually needs something a little bit cooler than body temperature to develop properly, right? So by suspending the testes in this scrotum, we're able to cool them a little bit, not having them physically in the body cavity. And we can even raise and lower them a little bit in that scrotum in order to carefully adjust the temperature that the testes are in. Okay. So as we produce our sperm in the testes, we're going to go from the seminiferous tubules, where we saw that cross section, and we're going to move into an area called the rete testis. So this is going to be as we start to connect up ultimately to our epididymis, we're going to go through rete testis and some little efferent ductules before we enter that epididymis, and then head into the vas deferens. So as we follow our developing sperm through the rest of the reproductive tract, so as we're going through the vas deferens into the body, we're going to pass by our accessory glands. So the first one we pass by are our seminal vesicles. Okay? So what the seminal vesicles are going to do is they're going to secrete an alkaline fluid. So in this alkaline fluid, we're going to have things like fructose, we're going to have enzymes, we're going to have prostaglandins. So this is going to change the quality, essentially, of the semen as it continues. So the sperm and the fluid that they are suspended in, right? As we go through the prostate, we're going to see that the prostate adds to that. It's going to add some more enzymes, and it's going to secrete some citrate. And then as we continue to head out of the body, we're also going to pass by the bulbourethral glands. The bulbourethral glands are secreting a kind of viscous fluid that contains some mucus. So all kind of changing the composition here. Okay. So when we think about the overall role of androgens in males, first thing we're going to be thinking about is stimulating spermatogenesis. And if we're earlier on in life, if we're still in this embryonic or fetal stage, sex differentiation. When we get to puberty, we're going to be using these same hormones to develop secondary sex characteristics. You'll see that androgens are also related to sex drive, to protein synthesis. They stimulate growth hormone secretion that affects bone growth in males during adolescence. And then, yeah, this last piece is diving back to that embryonic bit, the sex differentiation part. Okay. So we're thinking about sperm and their development and how we end up with functional sperm. We want to know something about the parts of sperm. Okay. So what we see when we look at a sperm is we break it up into approximately 3 parts. So we're going to have the head up here. You can see why we call it a head. Looks like a head, right? We're going to have a midpiece. So that's this part here. In the midpiece, we have a lot of mitochondria. And we have a tail, right? So here we have this whip-like tail, these flagella, essentially, that are going to propel sperm. You do want to break down what's in the head of the sperm a little bit further here. All right, so you're going to notice, all right, we have this region here, where we essentially have the nucleus of the sperm. So this is where our chromosomes would be carried, right? So this is where we're carrying actual genetic information. It's in this specific part of the head. And we notice we also have this little projection way up front. So this projection way up front on the head, this is the acrosome. So the acrosome contains a bunch of enzymes. These enzymes are going to be needed for fertilization. So basically, a lot of the physical structure of a sperm is in order to help deliver the chromosomes that are in the nucleus, right? We need the acrosome to actually effectively pierce into an egg and fertilize it. We need the mitochondria so that the sperm has energy and is able to swim. And then we need the tail to actually do the swimming. Okay, so the sperm are going to have to travel relatively far to actually fertilize the ova. Good. So to get to this fully functional sperm where we have the head with the acrosome, this mid piece with a bunch of mitochondria and a functional tail, takes a bunch of steps. Yeah. So we're going to start out in our basal compartment out at the edges of our seminiferous tubules. Okay. So this is where we're going to find our spermatogonia. Okay. So here, just when we saw this before, right, this was the cross section that looks kind of like that, and we had Leydig cells outside. Right. So we're looking at what's happening between two individual Sertoli cells, right? So this is a Sertoli cell. This is the neighboring Sertoli cell. Okay. So the spermatogonia stay up here. The spermatogonia are diploid cells. So that's what this 2N piece means here. And the spermatogonia can make more copies of themselves. So the spermatogonia can and will be producing more spermatogonia throughout the course of life, right? So they can do mitosis. They can make duplicate copies of themselves, right? So I want you to imagine one of the spermatogonia is just going to continue to do that, make sure that we have more spermatogonia to work with next time we want to make more sperm. Okay. Well, let's trace one of the spermatogonia. It's not going to do that. It's going to start helping us develop our fully functional sperm. Okay. So we're going to take one of these spermatogonia, and we're going to go through the process of meiosis, as we can see here. So through the process of meiosis, which you have hopefully covered in other classes, because we're not going to go in detail into the phases here in A&P. But essentially what we're doing is we're replicating our chromosomes, and ultimately we're going to chop up our genetic information so that we produce 4 genetically distinct haploid cells. Right? So we can see that as we go from the spermatogonia, in meiosis 1, we see that we're going from primary spermatocytes, which are still diploid, to secondary spermatocytes, which are now haploid, right? So we don't have double copies of all our genes anymore here. And then we pull those apart into spermatotids, right? Which are essentially the genetic information that's going to go into each sperm. So this is after meiosis 2. We've pulled apart all our genetic information into 4 separate pieces, and these are likely completely genetically distinct from each other. It's helpful so that we can scramble genetic information in humans so that we're all different from each other. All right. So you can see how we've passed between the Sertoli cells here as well. All right. So we had our primary spermatocytes able to travel kind of here across this tight junction, right? So they're able to go through these compartments, but other stuff wouldn't be. That's the point of the tight junction, right? And then this continued division is happening down here in this luminal compartment as we produce our secondary spermatocytes, our spermatotids, which we then package into spermatozoa as we start to gain parts, right? So our spermatozoa essentially look like sperm that we talked through on the previous slide, right? So we can see that they have a head, they have a midpiece, they have a tail, right? They're still essentially going to have to be kind of like activated a little bit later on, but they got their parts. Okay. So as we say, that's the process of spermatogenesis if you're looking at it from a picture perspective. From a words perspective, if we're words people, we're realizing that this whole process is happening from the basement membrane, so out at the outer edges, heading towards the lumen. Our spermatogonia can do mitosis, so you're going to think of one spermatogonium as staying to produce more so that we can continue to produce sperm throughout the entire course of life. But the other spermatogonium is continuing and heading towards sperm maturation. Okay, so we ended the last slide with our spermatozoa, right? So the spermatozoa, I think, right? We're down here at the end being released into the lumen. Okay, so those spermatozoa are going to get released into the lumen, so that hollow part of our seminiferous tubule. And they're going to stay there for like 20 days, right? And for those 20 days, they are immotile, so they can't move. They're not swimming around with their tails at this point. They're kind of hanging out waiting, okay? They don't acquire motility, so they don't acquire that ability to move until they get moved to the epididymis. And that happens by peristalsis. So that's why it's important that we have that smooth muscle layer, right? So we're squeezing these immobile spermatozoa to the epididymis with our smooth muscle and a little bit with our flow of luminal fluid. So that fluid that we know is coming from the Sertoli cells. And then they're going to gain this ability to move on their own. Okay. We are still going to help them with peristalsis though, because why not? All right, so they're going to be moved from the epididymis to the vas deferens, so that tube that heads back up towards the body. And it's really in the vas deferens where they're going to be waiting until ejaculation. So I don't want you to think that they're waiting for 20 days till ejaculation, this waiting period in the seminiferous tubules. That's actually part of the process of their maturation. The fully mature sperm are likely already in the vas deferens. Okay. So to release the sperm through ejaculation, we do need to control the sexual response. So this is going to happen through autonomic nerves coming from your brain, as well as some spinal reflexes. All right, so autonomic nervous system, remember, we've got our parasympathetics and we've got our sympathetics. So we usually think of that as rest and digest and fight or flight, not necessarily mapping on so well to the sexual response here, I will say. But in terms of phases of the sexual response, we're going to start out with the process of erection. So this process is largely mediated by the parasympathetic nervous system. So that rest or digest piece, that relaxation is going to allow the penis to fill with blood and therefore stiffen. Okay. And then we're going to go towards the emission phase and then ultimately ejaculation. We switch on to our sympathetic nervous system. So as we get towards the point where we're ready to actually release the sperm, we're flipping to the other half of the autonomic nervous system. We'll say the handy dandy mnemonic I was taught for this was point for parasympathetic and shoot for sympathetic if you are trying to remember which one comes first. Handy dandy. Okay. So the stimuli for this, right? Sorry guys, trying to be as clinical as possible, but also give you the tips and tricks. Okay, so stimuli for these. We got mechanoreceptors, right? So we can feel touch in the penis and in other erogenous zones. And we have stimuli coming down from our higher brain centers, by which we mean like your thoughts, right? Has a role to play here in these different phases of the sexual response. Okay. So here we have a nice clinical flowchart of the sexual response in males from your textbook. All right, so here we have our mechanical stimulation of receptors in the penis and our thoughts in the brain, essentially. So neural signals coming from pathways in the brain are our initial stimuli that get sent to the spinal cord. All right, so this is the info coming to your spinal cord. Ultimately, this info is going to make its way to the penis itself. All right, so the way it gets there, at first, we're going to increase our activity in our parasympathetic neurons that are going to the penis, right? So this is that process of erection that we're heading to. So these parasympathetic neurons, the way that they're causing the erection is that they are relaxing smooth muscle in your arterioles in this blood supply to the erectile tissues of the penis. So as we relax the smooth muscle, more blood flows there, essentially, is what we're thinking of. Okay. Blood gets kind of blocked from flowing back to the body, which is part of why the swelling continues to increase. All right, so we have this increase in blood flow to the erectile tissues, right, which means we've got more blood volume and we got more blood pressure here in those erectile tissues. All right, and as we see this blood volume and blood pressure increase, veins are small and floppy, so the neighboring veins get compressed, right? So we're blocking blood from returning to the body here. So this is one of the few places we see positive feedback in the body. Our big examples of positive feedback are usually reproductive or clotting when we think about A&P, right? So this reduction of the outflow of blood means that we are swelling even more, right? More blood volume, more blood pressure staying here. So that's our process of erection. Okay. So then from erection, we switch to emission. So as we go into this emission phase, this is a response to continued mechanical stimulation. And this neural activity is shifting. We're going from our parasympathetic part of the autonomic nervous system to sympathetic control. Okay. So what's the difference here, right? So we already have an erect penis at this point. Now we need to start moving the sperm and producing secretions to go into the semen. So they're not getting released yet, but they are getting made and moved, okay? So the epididymis is going to be doing some contraction, right? Vas deferens is doing some contraction, so is the ejaculatory duct, right? So remember this process of essentially peristalsis is part of how semen is getting moved, right? So fluids and sperm. The seminal vesicles and the prostate are secreting their fluids and their enzymes and adding them into the semen. And overall, this semen, the sperm, the fluid are all moving together into the urethra, right? So that's the emission process, is getting everything ready, essentially, moving stuff, but not out of the body yet. Ejaculation is the out of the body part, right? So sexual response continues, usually response to mechanical stimulation until ejaculation. So this is fully under the control of sympathetic activity. What's happening here is that we are stimulating contractions of the smooth muscle in the urethra itself, as well as skeletal muscle at the base of the penis. I think in lab, we haven't asked you to look at all the muscles within the penis itself, but they actually are there. They even kind of surround erectile tissues at the base of the penis. So these are going to start to contract during ejaculation. Urethra, smooth muscle is contracting. So this semen is going to come out of the body. Importantly, at the same time, our urethral sphincter is closing because we want to make sure that we're closing off like the pathway from the bladder, right? At this point, we just want semen coming out, not urine coming out. Okay, cool. We will end there for today, and we will pick up comparing to the female reproductive system on Wednesday.